Okay, so okay, so okay, so okay, so okay, so okay, so can you hear me?
Yes, we can. I can hear you.
Okay, very loud and clear.
Yes, it's very clear.
Okay. So our students are available currently. So we'll be talking about bioinformatics today. This will give us an edge over other students based on what is available in our own curriculum. So when we say bioinformatics, what do we actually mean? So for some of you that are in biology, biotechnology, uh, forensic science, science laboratory technology, biochemistry, microbiology. We look at bioinformatics from just like integrating biology and information. Integrating biology and information. So when we talk about that bioinformatics, we talk about a collection of data, analyzing complex biological data, such as protein information, gene information. For protein, say it's proteomics. For gene, we say genomics. So when we collect this data, we analyze them in order to understand or get an insight into biological functions. we term that study as a bioinformatics study. So there are different areas of bioinformatics study. Some of you must have seen areas in, for example, let me make this example. People in microbiology may decide to study or to know which organism is causing a particular disease in an area. For example, they will say Salmonella typhi or Salmonella or typhoid fever is predominant in Southeast.
Sorry, are you still there?
Yeah, I'm here.
Okay.
I'm here. So I'm trying to block every form of distraction. So for some of us in microbiology, might be working on Salmonella typhi, and you find out that the Salmonella typhi is actually, you go to hospital, they give you some drug, some medicine, but you're not getting well. So the doctor observed that most of the people that are coming in for treatment for typhoid fever are not usually, the drugs that are available are not really treating them well. What could be wrong? So the doctor might decide to isolate the organism, which is the Salmonella typhi organism, analyze the gene, After analysis of the gene, to get the DNA composition of the bacteria. And we have a database. When we talk about the database, talk about like a bank that houses information about biological data, such as proteins, nucleic acid, like the DNA, RNAs. So I say, okay, let's look at if the DNA in the Salmonella typhi is different from the DNA of the already established DNA of the Salmonella typhi. And they run, we call it a blast, and they blast the gene, which I'm having a reference gene of the Salmonella typhi. If the gene are not matching, we can now talk about why is the gene not matching? Are you sure we're not having a mutation? And that mutation might be as a result why the drugs that you're taking is not working. So that kind of study is a bioinformatics study. You're analyzing genomic data in order to study the cause of a disease. Sometimes I also say that, okay, diabetes mellitus is predominant in the southeast region of Federal Republic of Nigeria, or is predominant in West Africa. Like, for example, hypertension is predominant in Black Americans. So what could be in their gene that is making them more susceptible to developing hypertension? So somebody can actually look at, okay, let me look at the gene responsible for blood flow and the rest, or the gene responsible for blood vessel production and the rest. And they might actually get the gene and see that, okay, this gene, it will predispose you to hypertension. Even in breast cancer too, there are some genes that have been associated with the development of breast cancer. So if you go for early screening and you find out that you have such genes, you might actually need to start looking at other, early treatment or early, so that it doesn't develop into cancer. So that's what we look at in bioinformatics. People in forensic science will also look at bioinformatics in analyzing gene sequence. For example, in solving crime, you can actually use a bioinformatic tool in solving crime. We can also use bioinformatics to look at the genealogy of a family trace, or maybe let's say you want to know, there's this thing online where they say Igbos are the Jews, Igbos are part of the Jews. They might also want to say, okay, let's trace back the Igbo gene and know where the gene is actually coming from. So by that, we can actually trace the genealogy of an individual, know where you're from. You have seen some cases where two friends that never knew that they were siblings, two friends that never knew they were siblings. So I'm just giving a story so that we appreciate bioinformatics very well. Two friends that never knew that they were siblings. One of the friend daughter went and collected was looking for the father. The father doesn't know the father or the mother. He doesn't know his parents. So growing up, he got married and gave birth to children. So one of the child decided that I want to know where my father is from, and collected his father's DNA, analyzed his father's DNA, and put the father's DNA into a kind of website, or the DNA.com. I don't know, I've forgotten the name of the website. And so in that website, you can actually trace whose gene is closer to your gene. So when they did the screening and they found out that this particular friend of his have the same similar genetic makeup with his father. Or like, are you sure? Okay. They now talk to the friend and they went for further analysis, and the analysis showed that both of them are actually siblings. So that was how he connected to his lost family. So that's the power of bioinformatics. You might also be there. You don't actually know that maybe you might actually be a twin. But maybe during childbirth, maybe your twin is actually separated from you, and you just want to run your DNA test, and you run a DNA test and put it in the server. Before you know it, you can actually have a match. Somebody that you have identical DNA with, that probably must be your twin. For you to have the same gene makeup, the person is definitely your twin. You can also have maybe a lost brother, or a friend, like I just explained. So that actually gives us an insight of the bioinformatics. And there are tools that we use in bioinformatics to study biological data. There are tools that we use to actually study biological data. Hope we are getting the whole gist. I want to see your response. Let me see your responses and make your comment so that I can actually answer your questions. Make your comment so that I can actually answer your questions. So now one of the two that we'll be looking at today is pharmacokinetics. In pharmacokinetics, we study four basic parts of pharmacokinetics. We usually call it the ADME or the ADMET. ADMET, some will add T, that stands for toxicity. So the A is for absorption, which is how compounds enter the body, the systemic circulation. D is for distribution, how the compound is distributed, how the drug or how the medicine is distributed throughout the system. Okay. So M is for which actually occurs in the liver, and excretion, how the body metabolize drug, or how the metabolized medicine is eliminated from the body. So that's ADME. Now, prior to the development of bioinformatics, what we use in studying pharmacokinetics are animal models, call them animal models, where we take a rat, for example. We have the albino rat, the most musculus domesticus. We have the Novecus, different rat model of guinea pigs. We use them to study. M is for metabolism. We use them to study pharmacokinetics, how the drug is metabolized, how the drug is absorbed, how the drug is distributed, how the drug is excreted. So that is pharmacokinetics. So before the development of the bioinformatics, we use animal models to study this. So you must have seen some final year students buying rats or using rats for their experiments. So that's what we do, use rats for the experiment. And those experiments can be translated into human studies. But we don't just rush into human studies, we use animal studies to get to a particular point before we can translate into human studies. So one of the things we do is use the animals for pharmacokinetics. But in recent time, we don't use animals again because the animals also have rights. We don't use the animals again, they also have rights. And the animal rights must be preserved. So that's why we're actually using a bioinformatic tool called the in silico pharmacokinetics. So in the in silico pharmacokinetics, I don't know if you're seeing my screen. I don't know if you're seeing my screen.
I can see your screen. I can see it.
Our audience on YouTube.
Okay.
Yeah, if you can see my screen on the YouTube channel, make a statement. Okay, saying is not clear. Okay, I can say it. So what are you seeing actually? I want to know what you're seeing. I want to know exactly what you're seeing. Okay, so I want to know what you're seeing. I want to know what you're saying. Okay. 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 So yeah, we are on the same page now. We're on the same page now. I'm also seeing. Okay. Yes, we're on the same page now. So the ADME process include metabolism for M, A for absorption, E for excretion, D for distribution. But most time we also added T, which is for toxicity, to know if the drug is toxic or not, to know if the medicine is toxic or not. So now, for example, let me give us a typical one. You all know the drug Lonat, right? We all know the drug Lonat. We all know the drug Lonat, right? Now, Lonat contains two active drugs, lumefantrine and artemether. Lumefantrine and artemether. Now, these two drugs, usually most of us don't take it the right way. Most of us don't take it the right way. How do we take Lonat so that we'll take it the right way? If you look at the leaflet of the drug, they will always tell you, take this drug with a high-fat diet. But most of us will just take the drug and open our water, drink it with water, and that's it. Not knowing that we have to take it with the high-fat diet. So now let's look at how we can do some pharmacokinetics. Let's look at okay. Just hold on a little. So most of us don't know how to take the LONAT. Okay, so okay. Can you hear me? Uh, this is echoing. Okay. Uh, can you see my screen? Okay, I'll type Swiss admin and search. You can see the Swiss admin. Click on the Swiss admin. Can you hear me now? Can you hear me now? Okay, so this is the Swiss admin. And for me to check the bioavailability of the pharmacokinetics of a drug or of a medicine, you can type, there's what called the canonical smile. There's what called the canonical smile. So you paste it there. Let me just show you. Let me go to the basics. How we get it. Type in PubChem. Type in PubChem. That's the first thing you do. PubChem is a database that houses thousands of chemical compounds. So click on PubChem. Click on PubChem. It's a database that houses thousands of chemical compounds. So when we click on PubChem, if there's any medicine that you want to check the pharmacokinetics of the medicine, you take it to PubChem. So let's take lumefantrine. Let's take lumefantrine. I've typed lumefantrine. Yes. Somebody asked canonical smile. Yes. Somebody asked canonical smile. Yes. So I've typed lumefantrine. I'll click on the lumefantrine, or I'll just go to the smile. Or I'll just go to the smile. It's loading. It's loading. So go to names and identifiers. You see the IUPAC name, the smile. You copy the smile. Copy it, copy. Then take it to the Swiss admin that we have opened. Paste it there. Then give a space. Give a space, then type the name. Fantrain. We can actually search for more than one compound. Let's search for gentamicin. You know, most of you, when you're not well, you're having one sickness or the other, you go to the hospital, they'll say you give the person gentamicin injection. Well, you were wondering, doctor, why are you not giving me gentamicin tablets? I'll explain to you why gentamicin doesn't have a tablet form. It's only injection. The light, you said you're lost. Tell me, just write it down for me so that I can actually explain, carry along. So you copy the canonical smile, you copy it, then let's go back to our Swiss admin. You paste it, Control C. Control V rather. You paste it. Then this is gentamicin. So let's say we do this two, just this two. You click on run. Okay, now this is the first thing I did. Since you are looking at the pharmacokinetics or you are trying to study the bioavailability of those medicine, the first thing we did was to go to PubChem, where we have thousands of chemical compounds, medicinal compounds that are stored. So the name of the website is PubChem. Once you click on PubChem, you see the search bar. You search for any compound, any drug, any medicine, or any phytocompound that you're looking for. You get the phytocompound. You go straight to the canonical smile. Copy the canonical smile, or let's just say the smile. Copy the smile. After copying the smile, you come to the second web this that you open, which is the Swiss admin, and paste it on where you have the list of smiles. Paste it there. You can have hundreds of smiles there. You paste it and give a space bar, type the name. So that when you're running your this thing, your work, it will show the name there. You can see that it is showing the lumefantrine. What are you actually seeing? What are you actually seeing on the screen? What are you seeing on the screen? Tell me one thing you're seeing. Just mention one thing you're seeing. Okay, lumefantrine. What else are you seeing? What else are you seeing? Francis. Okay, so for those of you that came late, the video will still be on YouTube so that you can actually go back and watch the video. Video will still be on YouTube. You can go back and watch the video. So you can see lumefantrine. On this part, you see on the pharmacokinetics, you see GI absorption. GI means gastrointestinal. GI absorption low. That means if you take it orally, okay, comes on, okay. We're on the same page. We're on the same page. So if you take it orally, it means that you will not get the best of the drug. That's why you see GI absorption low. You will not get the best of the drug if you take it orally. But now the people that produce the drug decided that, okay, what can we actually add to this drug that will increase the absorption so that you get the maximum benefit of the intention of the drug, which is to treat malaria. They now discovered that, okay, this drug is highly lipophilic, so you can take it to the high-fat diet. So that if you're not taking the high fat diet, the high fat diet will now facilitate the absorption of the drug. Now let's go to gentamicin. For gentamicin, you'll see that GI absorption is low too. GI absorption is low too. So taking the gentamicin orally will not work. So that is why we only have gentamicin in the injection, which is either through intravenous or through intramuscular. So that is that for pharmacokinetics. Another thing you can see there, is the boiled egg. It's the boiled egg. It's all called boiled egg. You can see show boiled egg. When you click on show boiled egg, it will show you this structure that looks like egg. Now, if you say show molecule name, you see that the molecule is not even there. The molecule is not even close to the egg white and the egg yolk. You can see the egg white and the egg yolk here. So the molecule is not there. But if we search for drugs that have good oral absorption, let's assume you are looking at, for some of you that take ciprotab, let's look at ciprofloxacin. Let's look at ciprofloxacin. Let's look at ciprofloxacin. You can see the smile. Let's copy the smile. Let's copy the smile. Then go back to our Swiss admin. Paste the smile. Control V. Cipro. For some of you that take Cipro as your drug, let's look at what Cipro will give us. Ciprofloxacin is an antibiotic. An antibiotics. It's an antibiotics. Okay, so okay, so you can see Cipro. For those who are saying that it is not clear, you actually need to change your settings. But actually need to change your settings. If your pixel is low, it will be blurry. So you need to increase your resolution either to 420 or 7 something in your YouTube. So you can see that the Cipro, for Cipro, ASU is on strike. So the light, did it come in late? So we have GI absorption for ciprofloxacin, which is very high. You can see it, high pharmacokinetics. You can see the high pharmacokinetics. High pharmacokinetics. So that means if you take ciprofloxacin via the oral route, you have a very good absorption, which will meet the pharmacological drug is to help treat bacterial infection. So if you take this ciprofloxacin orally, it will perform its job. But for gentamicin, you have to take it via oral route. That is not the oral route. Okay. I'm trying to expand the screen so that you can actually see better. Can you see? GI absorption high. Can you see I've expanded the screen? GI absorption is high. Delight, go to your settings. Go to your settings. Okay. Okay. So GI absorption is high, which is gastrointestinal absorption is high. Then you can see BBB permeant. BBB permeant means blood-brain barrier permeation. Blood-brain barrier permeation. So if it is no, it means that the said drug or the said medicine cannot be used to treat neurological disease or neurological problems. But if BBB is yes, it means that the drug can be used to treat neurological problems. So you can see this is the structure of ciprofloxacin. This is the structure of gentamicin. Gentamicin. And this is okay. So if I click on boiled egg, show boiled egg. If I click on show boiled egg, I will have something like this. You can see the boiled egg. Show molecular names. You can see Cipro there. You can see that it is only Cipro that is showing on the egg white. If the drug can actually, if the medicine can actually enter the brain, you will see it on the egg yolk, which is the yellow portion. If the drug cannot enter the brain, you will see it on the white portion. But if the drug cannot be absorbed at all, you will not see it either in the yolk portion or in the egg white portion. You will see it somewhere here. You can see this one here. You can see this one here. This blue, this is one of the drugs. One of them did not even appear at all. So that's what we do in pharmacokinetics. That's what we're doing for pharmacokinetics. So another thing we can actually look at again is to look at the protein structure. To look at the protein structure, to determine the protein structure, and for example, we can actually use post-param actually use post-param we can actually use post-param to study the physicochemical properties of a protein. So let's assume we are using this letter here and paste it here. Ctrl V, compute parameters. If we compute parameters, it will show you the amino acid composition of a protein. For example, how many of you can determine if a protein is made up of mostly essential amino acids and non-essential amino acids? Say okay. If I ask you, make sure you eat proteins are available. Make sure you eat proteins that are available. Or for example, they'll ask you this protein, this protein, this protein. Which of this among these three proteins do you think will be more nutritious? Which of these proteins do you think will be more nutritious? So how do you know the protein that will be more nutritious? Who can give me an overview on this thing on the comment section? Now let's look at 3 proteins, for example, or let's look at 2 proteins. For me to look at the 2 proteins, there are 2 websites that we must go through. We have NCBI search. NCBI. Let's go to NCBI. Okay, no, this is BLAST. Let me just go to NCBI. National Library of Medicine, National Center of Biotechnology Information. In NCBI, I'm interested in 2 proteins. One, the protein collagen protein. Search collagen protein. You click on conserve, click on proteins. Click on proteins. I'm looking at collagen and albumin, 2 proteins, collagen and albumin. Collagen and albumin. Okay, this is collagen in Drosophila melanogaster. Let's see Gallus. Okay, let's take any of the collagen. You click on FASTA. FASTA is the amino acid sequence. FASTA is the amino acid sequence. You can see the amino acid sequence here. Yes, you can see the amino acid sequence here. Let me copy the amino acid sequence. I'm copying it. These are the amino acid sequence. Each letter represents an amino acid. Each letter represents an amino acid. Now let me go to the and paste it here and compute. I just computed for collagen. For collagen, you can see that the amino acid composition for collagen, we have alanine, 6.6%. We have glycine, 17.0%. We have proline, 11.1%. Now, how many of you know the essential amino acids? How many of you know the essential amino acids? Yes, the video will be downloadable. The video will be downloadable. Video will be downloadable. So how many of you know the essential amino acids? You know it, somebody say you know it's more. Now look at this one now. Now look at this collagen. Look at this collagen. You find out that most of the essential amino acids are not there. The amino acid composition, you can see that proline is not an essential amino acid and it is very much there. Glycine is not an essential amino acid and it is very much there. Phenylalanine is an essential amino acid. Threonine is an essential amino acid. It's also very small there, 5.1%. Tryptophan, an essential amino acid, is 1.3. 1.3. So let's compare. You now see if you're giving two protein, collagen and albumin, and they are to select one that will be more nutritious. Now let's go to let's search for albumin. 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 You click on the FASTA. FASTA will give you the amino acid sequence. Homo sapien, which means this is human. Homo sapien. Okay, the light said essential are the ones that we gain from food. Well, of course, you're correct, but we define it better as the amino acid that our body cannot produce. Therefore, they are required in our diets. They're required in our diet. So you can actually take them through diet. Our body cannot produce them. So we need them, we need to take it from food. I think it's not downloading. I don't know. Okay, so we now have it. We now have the FASTA. Let me go to my. Let me just reset. Control V. Compute parameters. You can see that we have leucine, lysine, and essential amino acid is even 10% of the total constituent. We have valine. We have thyroxine, which is a conditional. We have tryptophan. We have threonine. We have threonine. We have lysine, an essential amino acid. We have leucine. We have isoleucine. So which one do you think will be more beneficial to our body? Of course, albumin will be better than collagen. Albumin will be better than collagen. So you can see the number of amino acids, 494. You can see the theoretical isoelectric point, which is 6.85. So at 6.85, this protein is neutral. That's the meaning of isoelectric point. Somebody says, "Sir, why was the FASTA hard to find?" It wasn't hard to find, just network. Yes, you're very correct. The light, you're very correct. To know the protein that will be more nutritious, we need to check the amino acid composition. So it is the amino acid composition that will determine if the protein will give us essential nutrient or not. You don't take a protein, somebody just say, okay, this is protein. And because the person just said this is protein, you don't say, okay, this is protein, start consuming. No, we need to know if the protein is made up of more of essential amino acid or not before we can actually determine that this protein is more nutritious than the other. So I love that. Do you have any more questions? The next thing we'll be talking about is molecular docking. Molecular docking. The next thing we'll be looking at is molecular docking. Now, what is molecular docking? We want to determine, yes, molecular docking. To determine the interaction between a molecule, which is sometimes called a ligand, and a protein. So we are looking at the interaction between the ligand and the protein, looking at the bonded and unbonded atoms, how the ligand interacts with the protein, how the ligand interacts with the protein. So this is just an overview. I will not want to dwell much on it, just for you to have an insight that such things are applicable. You must have heard that enzymes catalyze substrates. Enzyme catalyze substrates. So when we say enzyme catalyze substrate, the target is the enzyme. The ligand is the substrate. Sometimes the target might be a protein or an enzyme. The ligand usually can also be a protein or an enzyme, or more especially, ligand is usually a small molecule that can fit into the active site of the protein or the enzyme. So at the end of this, I will post a link. The link will contain most of the, you can actually go to that link and watch most of the videos in order to have a broader understanding of what we are doing. So I'm just explaining the concept of molecular docking, just the interaction between the ligand and the protein. So what I just, the link I'm sending now is a step-by-step, the step-by-step procedure for molecular docking. The step-by-step procedure for molecular docking. Okay, I also get a link for pharmacokinetics. I also get a link for pharmacokinetics. So the videos, the link for pharmacokinetics will also be posted here, so that you can go back, watch the videos, and so that you can actually prepare well for things. So I'm sending the full playlist. So I've sent the full playlist for bioinformatics. So you can actually look at the full playlist, understand it better so that you can actually prepare well for your defense. Thank you. We'll see you some other time. Okay, Samuel, when I ask your question, please. Yes, best. Please watch the playlist. You will get a full understanding. You're welcome. BBB permeance means blood-brain barrier permeation. Blood-brain barrier permeation.